Hi, good morning, everybody. Welcome to the last day of the KLF Literature Festival. Today we have an early session at 9:30. The normal start time I know is at 10 a.m. But today to accommodate Dr. Sekar, who just landed here today morning and will be leaving in a few hours, the only slot that we could get was this an early slot, 9:30 a.m. But for the few of you who have been able to make it here, I'm sure you will enjoy this session. So my job today is to briefly introduce Sekar to you. Sekar is from the state of Tamil Nadu. So a Tamilian, but he still has a Malayali connection. His wife is from Kerala. So when he also reminds me often that we all Malayalis are also from Tamilians originally. So Sekar for the last 25-30 years has been in the US. He did his basic undergrad up to undergrad education here in India. Then went for his PhD at the University of Georgia, did his PhD in biology. He was mainly focusing on virus. But then after this PhD, he joined Genentech. Genentech is in San Francisco, in the Bay Area of California. Some of you might know, Genentech is the first biotech company in the world and one of the largest. But now they are part of the Roche group. So Sekar has been in Genentech for the last, was in Genentech for the last, for about 21 years. Then in last year, he quit Genentech and has started his own company in San Francisco. That's a company that is developing new molecules for cancer treatment. Sekar has a great passion for India and science education. So in the last many years, he has been coming continuously to India, at least 6 or 7 times a year, and working with various organizations like ours, MedGenome, SciGenome, and other companies and hospitals in trying to push science, and also trying to ensure how science gets applied in the right areas in the right way. So though he's a cancer scientist and one of the world's famous cancer scientists with numerous publications in all well-known science magazines like science journals like Nature, Nature Genetics, Cell, and all those, he has, like I said before, a great passion for all science. So he's done a lot of work in many plants, various animals, and finally the cobra. So that has been completely a project which he conceived right from the beginning and managed it exclusively with a small team of Indian scientists from the beginning to the end. And the great recognition that they got was that this month in January it got published in Nature Genetics and also got into the cover of Nature Genetics. So getting the Indian cobra and their paper in the cover of Nature Genetics is a great achievement for them and also for us as Indians. Right, some work from India, because along with this new company that he started, he also became the president of the SciGenome Research Foundation. This is a foundation in Kerala set up to promote science education and research. So a paper from SciGenome Research Foundation led by Sekar gets into the cover of Nature Genetics. I think it was one of the most satisfying moments in my life to see that happen. And I'm sure it's just the beginning. There will be a lot more papers coming up. Sekar has been working very hard on a lot of venomous animals, different snakes and many other venomous creatures. Maybe he'll touch upon it briefly today, why it's important and what are we going to get, not just science learning, but how are those going to be applied to practical use and what is the benefit that we humans will get out of understanding the genome and the genomics of these organisms. So I will turn over the stage to Sekar, and Sekar will take you through the Cobra Genome, how they did it and why they did it and what more we can expect from that. Over to you, Sekar.
Yeah, thank you, Sam. That was the longest introduction I've had in my life, okay. If you feel like and you don't want to struggle on the stage, you've also tried to tell you why we study and the fascinating things about snakes, but also biology itself in general. And I'm glad to see some students in the audience as well. So let's start I mean, yeah. So I think this slide to your right, you know, everybody in this room, you know, if unless you were cloned somehow, you know, all of us start out as a single cell, right? A fertilized egg. And if you take a fertilized human egg, it basically always reproducibly turns into a human. You can take a mouse egg, at least in our lifetime, it'll always be a mouse. Same as with a grain of rice, if you put it in the soil, it's going to be rice, and maybe the same with corn. So inside each of these root cell or the egg cell is all the code that you need to actually make that whole organism. It's actually pretty profound. And the egg, the human egg, is the size of a pinhead or even smaller, right? You literally have to look at it under the microscope. And upon fertilization, it just, you know, for 9 months, there's a beautiful developmental choreography that happens. And people have been studying and trying to understand what and how this happens. And so the first question is, what is the code inside the cell? And where is this code sitting? And that code is essentially in the DNA inside the cell in a compartment called a nucleus. DNA is the chemical. I don't know, many of you heard of DNA? How many of you heard of DNA? There you go. So it's the DNA that's doing all of this, right? And so that's the fundamental code. So starting in about 1970s, early 70s, there's been an enormous effort to figure out the sequence of DNA inside various sorts of cells. You might have heard of the Human Genome Project that happened in about 2002. And the goal of it was to figure out the coding sequence that makes us who we are. What is this blueprint? What makes us human? And it took 10 years, about 16 countries, and we spent about $3 billion to get the first reference genome. And Sam here earlier was talking about various things. He and his colleagues in India have transformed the application of DNA in the context of medicine, particularly human stuff, right? And so what did the Human Genome Project try to do? So here we are, on your right, on your left is a little cartoon of a human, and if you go and look inside a cell, the DNA is organized in chromosomes, and at the bottom is the chemical that makes up the chromosomes. This is what people have been trying to map, and in fact this was mapped, completed in about 2001. We have the complete sequence of the human reference genome, and now we have many, many other genomes. And what can you do with this information? So if you simply look at DNA, if you took a novel, right? If you took a storybook, a novel, it's made of words, and the words are made of alphabets, and the alphabets and the words put together as sentences. Sentences organized into paragraphs, paragraphs into sections, and then you put the whole thing together, you build a novel. The information of the cell is no different. It is exactly the same sort of thing. You have a large, the alphabets of the DNA world are 4 letters. They're organized into a string, and they have a meaning. And the cell's machinery is able to figure out, ultimately, to make the correct proteins from that, and that's what allows us to be who we are. So, for example, you'd have heard insulin. How many of you know of insulin? Okay, what is insulin? Insulin is a protein, and you always associate it with diabetes. And all of us make insulin in our body, but the code needed to make insulin is actually in the DNA in the cells in our body, and that's how we make insulin. So when Human Genome Project started out, this is what they were trying to do. The human genome is organized into 23 pairs of chromosomes. I told you there are 4 alphabets, A, G, C, Ts, and it took 10 years and approximately $3 billion to figure it out. And this is a remarkable achievement. You know, you can, we always make a joke saying, hey, this is not rocket science, in the sense when we say a problem is not that hard. But I would say the sequencing of the genome was a rocket science, and it took a lot of people and a lot of money over time. And what can you figure out using it, right? In fact, if I told you all of us originated in Africa, out of Africa hypothesis, the modern humans left Africa about maybe 150,000, give or take another 20,000 years. We moved and came down, and India is one of the spots where a lot of things that happened as well. But how do you figure out how we are all related? If I sample the DNA from people in this room, we can understand very quickly how related we are based on the composition of the DNA. And any two individuals in this room will differ in our DNA sequence. There are 3 billion characters. We will differ in about 1%. The more closer we are, the more related we are. The more similarity in the DNA sequence. So using this, people have done a lot of stuff about migration, your ancestry, and then you can go and apply this in the context of medicine. There's a little kid with his mom, a beautiful picture, but he was in the hospital probably over 100 times by the time he was 6. Medicine could not, the modern medicine at that time could not solve his issue. And DNA sequencing fixed his problem. They figured out what the issue was by looking at the code, the blueprint, right? Same here. There are 2 kids who are basically running, really teenagers. That on your right, that's them at 5 years of age. They basically had neurological problems. All they had to do was sequence them, and when they did, they found 2 changes in their DNA, and they immediately knew that these proteins that are getting screwed up could be fixed very easily by giving them 2 drugs that were already in the market. As soon as they started putting these kids on the right drug, you cannot tell a difference between them and any other normal child compared to that picture on the right. So this is what DNA science is beginning to do. It's a sort of revolutionizing modern medicine. So if you go to a doctor's clinic, you'll take a temperature, you'll get your blood pressure, they'll look at your eyes, you'll stick out your tongue, all the vitals, right? But underneath all of that is also DNA information that's now percolating into modern medicine. So a combination of genetic medicine combined with the traditional medicine is going to change the way medicine is applied and how it's used to treat people. So with that sort of background, the one take-home message that I want you to keep in mind is DNA sequence is a fundamental blueprint, and that sequence tells you a lot of things, right? Fast forward, and for a minute, let's think about venomous animals for a second. Okay. This is Kerala. I don't need to tell you guys that this is probably one of the most biodiverse parts in the world, particularly if you go to the Western Ghats, right? So how many of you have seen snakes? There can't be anybody in this room that's not seen a snake, right? Okay. How many of you have been bitten by a snake? Okay. Have you known of anyone that's been bitten by a snake? Okay, there you go. Okay. So snake is something, sorry, you were about to say something? Sorry, I didn't hear you. No, but go ahead. I can't hear you. Okay. So the point is, snakes are fascinating, but there are about 3,000 species of snakes. Maybe about 600 are actually venomous. The other 2,400 or 2,400 snakes, they're not out to bite you, or their venom doesn't hurt you, right? So here is a little chart of all the venomous animals, or at least the major ones, major groups, in what we call a phylogenetic tree on your left. And then on the right are some pictures, right? Okay. So as you move up the evolutionary chain and you come up to mammals, mammals are not generally venomous, right? Humans, if they bite somebody, we're not injecting venom. But there are exceptions. There is something called a slow loris and platypus, 1 or 2 other things that are venomous. But majority of the venomous animals are in the reptilian and a lower clad of evolutionary chain. But the estimate is we have about 2 lakh or 2.5 lakh venomous animals. And we have studied only a very, very, very small fraction. And even within that, we have not understood the DNA blueprint. I told you, humans, we sequence, we got the blueprint. A snake, some snakes lay eggs. Others actually give birth to babies, but they do make eggs, but they keep them inside. But the point is, if you take a cobra egg, you'll get a cobra out of it. You take a python egg, you get a python out of it. How? Why? Right? So, and when snakes bite, they inject venom, obviously. And when they inject venom, their goal is, humans are not their target. We are an accidental victim because of accidental contact, particularly people working in the fields. High density of population in the rural areas where these conflicts occur. Snakes mean no harm to humans, and they're not interested in us. They want to go away. And they're extremely important in the ecology to control rodents. If snakes are gone, we will have so many rats and mouse and stuff running around. So without them, we'll be actually worse off. So, but at the same time, snake bites get so much of publicity because very often lead to a lot of serious effects, but also death in many cases. We'll come back to that. But what is venom? Venom is a lethal cocktail of proteins, some other lipids and other chemicals, and it's a mixture. And what does a snake do? There are different types of snakes, different types of venom. The general principle is when they bite their prey, be it a mouse, a rat, whatever, they want that rat to be immobilized and they want it to die as quickly as possible. And they consume the rat or mouse slowly, and even as they're consuming it, they're starting the process of digesting that animal that they struck. So, how often do you think does a snake eat? Any idea, generally? How often does a snake eat? Any guesses? I can give this microphone down if any of you want to try it. I think we can make this a bit more interactive and fun. Otherwise, yeah.
Once in a week.
Once in a week, okay. Any other guesses? Once in 2 weeks, yeah. So it depends on the snake. It can be once a week. It can be once in every week. And there are snakes in certain islands of China that eat only 2 times a year. These guys are amazing adaptation. And snakes are essentially lizards. They went underground in the evolutionary time scale, came out legless, and some acquired venom glands to catch prey. And the venom gives them a certain advantage in capturing and eating the prey. But if you were to be attacked by a python or an anaconda, other things, they use their muscle to constrict the prey and eat them, right? But this venomous snakes, their cocktail is a mixture of amazing set of proteins or peptides that have been evolved over time to do various things. So, for example, when a snake bites, and if you're put in a hospital and observed, you might get slurred speech, your breathing might slow down, your blood pressure will drop, you might have palpitation. There's so many things happen. But I'll give you one example how to relate the venom to the potential application. When a snake bites, your blood pressure drops. So there is something in that venom that causes the blood pressure to drop. So if you figure out what it is and isolate it, it can itself become a useful drug. In fact, that's been done using Brazilian pit vipers, where a particular peptide from the Brazilian pit viper was studied and evolved to create the most widely used blood pressure medication today. And so there are a lot of applications of these venomous animals, but without boring you, there is a bigger problem and a bigger crisis to solve in India, and it's a socially relevant issue, and we'll come back to that. So what I was telling you was we can do a lot of things. We can learn biology, we can look at potential drugs, and the more important and immediate and urgent need is anti-venom. Okay. So this is what I told you already. I'm not going to bore you again with this. This is the beautiful world of snakes, and they're related to lizards. There are about 3,000 odd species. Only about 600 or so are venomous, and they're absolutely fascinating in terms of adaptation and what they can do. Some climb up the tree, some are on the ground. It's a ridiculously diverse and beautiful set of animals. and they instill a fear in humans for a very, very evolutionary reason, and we run away from them and they run away from us. But this is a crisis that I was telling you about. So if you look at the map of the world and ask, where do snake bites occur? Which part of the world is it most common and most devastating? India is the capital of snake bites. We tell of diabetes being this capital. Equally, we are, because we have an amazing diversity of snakes. I'll tell you, there are 4 big snakes, and they cause over 50,000 deaths a year in India. There is over, in the world, about 125,000 deaths. India accounts for about 46,000 of those. Okay, well, death is one side, it's pretty bad. But when you're bitten by a snake, the disability caused by them can have enormous economic consequences for families. And in India, annually about 130,000 or 1.3 lakh people get disability related to snake bites. It's a serious issue. So what do you do about it? How can you solve this? How can you treat people bitten by snakes? Yeah, there is technology, and people do this the following way. So you might have seen, if you ever went to a snake park, they will extract a snake venom. They'll hold the snake by its head, put the fangs on a little funnel. The venom will come to the bottom of the bottle. Then you simply take it and put it into a horse. A horse is a very large animal, and so it can make antibodies without killing itself. And so these antibodies, essentially, when you go get a vaccination, I'm sure, I think maybe, I don't know about the younger generation, but many of us in this room would have gotten a polio vaccine, a smallpox vaccination, other things. When you get vaccination, you're getting immunity against that particular bug, and that's all because a type of cells called B cells start making antibodies. Horse also has the same type of immune system, B cells, and so when you put the venom, it makes lots of antibodies. People then purify the antibodies and then package it into an anti-venom, and that's what's given to people. There are serious problems with this. The technology was invented in 1895. Incidentally, the first anti-venom this way was created in Pasteur Institute in France against the Indian cobra, because French were all over in various parts of the world, so they needed an anti-venom. And this horse, when it gets a cold, it's making antibodies against the virus. When it gets another type of infection, it's making antibodies against that. Yeah. So we take all these antibodies, and then you essentially are putting it into people. You're putting a foreign protein. And so if you are bitten by a snake and you go to a hospital, they will not, if you're lucky enough to get to a hospital where there's anti-venom, they will not give you an anti-venom until you start showing symptoms. That's because the horse anti-venom can kill you. Only if it's absolutely needed, they'll give it to you. Otherwise, it is going to create an allergic reaction so severe in some cases, the patients die of the anti-venom, not of the snake bite. Anyway, so this is a 100-plus-year-old technology. But the field, I talked to you about insulin. The world has changed in the context of insulin, and this was how insulin used to be made in the 1920s. You went to a slaughterhouse, collected the pig pancreas, ground it up, purified the insulin. It was not great, but it was as good as you could do, and that was because prior to the 1920s, if you got diabetes, you're dead. It's a death sentence. There was no cure. But came along insulin, it changed the world. But this was still not produced recombinantly. You still need a lot of pig pancreas. And so in about 70s, colleagues at Genentech cloned the insulin gene. Eli Lilly commercialized it. They can make it in E. coli. E. coli is a bug that colonizes your stomach, but it can also be told to make recombinant proteins. That's where you get the human insulin today from. So what we proposed to do when we started this project was, could we address the snakebite crisis by using technology? So we started, there are these 4 big snakes in India, and we started by sequencing the Indian cobra. These 4 big cost 50,000 deaths. So we have completed sequencing of that. And essentially, this is how the DNA is packaged in 18 chromosomes, and it uses a ZW sex chromosome system. We applied a number of technologies, including being able to cut a single chromosome and sequence it. And when we did this, we got a very good assembly of the DNA. And using that, we could actually predict all the protein coding genes. So this circle outside represents each chromosome, and it's telling you, lots of pieces of information. But for our discussion, it's not that important. And what can we do with it? We can zoom in and start asking, where are the venom gland genes? What are the genes? What are the proteins in the venom gland? Now we have the blueprint. From the blueprint, we can actually learn a lot of things. Okay, how many of you think which one is the snake in this picture? The one to the left, one to the right, one in the middle. There are 3 pictures in the bottom. There's all 3 are snakes, all 2 are snakes, so the one in the right, okay, I'll tell you all. The rightmost one is a lizard. What is the one in the middle? Okay, it's also a lizard. And the other one is, on the left is a snake. But you know what happened during evolution? Snakes lost their legs, I told you, right? Lizards are older than snakes, so that's where at the other end, as you move down the evolution, there are DNA changes that were showing on the top that led to this loss of limbs. So we can learn a lot of little things along the way. So we'll skip this one in interest of time. So we were able to take different organs from the snake, including the venom gland, and we were able to figure out what genes are specifically expressed in the venom gland. Venom gland is essentially a co-option of the salivary glands. And so we figured out a number of proteins, and ultimately what did we want to do? We wanted to come up with, hey, what are the most important proteins in the venom that are causing all the physiological effects that we see when a snake bites. So once you have the blueprint and you know which proteins they are, you can actually, this is what we call the venom cocktail book for the snake. It doesn't need it, it's more for us saying, well, these are the proteins in that venom that's causing the physiological effects like drop on blood pressure, preventing blood clotting, and so on. So we took all of that information, and we can now go from this 100-year-old technology to something called a genome-guided 21st century technology. So we started the DNA, we get the sequence, and we figure out which ones are the toxin genes. And I told you, if I can make one protein, insulin, using recombinant technology, I can make 10 proteins using the same technology. So you can make each of these proteins, and then you can make antibodies using synthetic antibody technology. It's not beyond us. This technology has been around for the last 40 years. And so we propose that we can make synthetic anti-venom that are safe and also easy to give to people, and more widely available, and cost-effective and affordable, and safe. And that's where we're heading with all of this. And we're sequencing many other snakes and many other venomous animals from the evolutionary timescale. How long do you think jellyfish have been around? Any guesses? 100 million? 500 million? Yeah, you're about right. It's about 600 million years. And you come to the other end, snakes are only about 100 million years. And spiders, scorpions, there's certain types of cone snails. There's an amazing array of stuff to learn from nature. And each one of these tell you a lot of interesting things. Anyway, that's what we're trying to do. But our important goal, immediate goal, working with a number of people, both here in India and our collaborators in Canada and Germany, is to actually see if we can produce an anti-venom, first for the cobra, and then we'll start doing the other snakes, and we can combine them to create a universal anti-venom that will make snake bite related disability and death a little bit of history. It's a shame we haven't done it already. I think we can, and so that's what we're going to focus on. So I'll stop here. There's a number of colleagues of mine that helped. In fact, some of the cobra specimens came from a Kentucky zoo in the U.S., and then I have colleagues, at the Kerala Forest Department and others that have helped enormously with this. So I'm going to stop here, and I'm happy to, if there is time, I'm happy to take 1 or 2 questions. Yeah, no problem.
Technology helps in developing anti-venom. Is the same anti-venom applicable to all venoms, that is cobra, the other snakes and all?
Yeah. So yes and no. So the issue, I think, is there's going to be some variation in the components, venom components. So you'll have to probably say, for example, these big 4 snakes in India, the crate and the cobra are very related. Most likely it might work. but we still have to get the information on the crate and figure out. And you can keep adding antibodies. You make one against Cobra, King Indian Cobra, and then you have few more proteins from the crate. Exactly. And the technology exists to figure out the sequence, the blueprint, and it's not that expensive. So we should really do it. I mean, there's no reason to go, with our eyes blind and try to find a treatment, right? Yeah, I think there are many vaccines or other colleagues in India working on anti-rabies as well. But I think the most important thing there is, I think we also have to vaccinate the dogs in the streets, right? And that's a big vector, apart from other rodents and other stuff. But that itself will help. There is a big campaign through Blue Cross, Blue Shield, something in southern Tamil Nadu and all, but I don't know about here in Kerala, to vaccinate the dogs. More than humans to start with. Yeah. Okay, thank you.
Thank you, sir.